Hello students. So today we'll be discussing of a very important topic, which is the protein metabolism. Specifically, our learning objectives for today will be these things. One thing, general features of proteins, how the proteins are digested to amino acids and how the amino acids are absorbed. Then we are also going to see what are the general reactions of amino acids or the protein. What is ammonia and how it is neutralized in the body. So that also we are going to see. Then how the ammonia is disposed of in the body, which is specifically we are going to learn about the urea cycle. And then we are also going to learn what are the disorders related to the same. Okay. So moving ahead, we all know that the proteins are nothing but the polymers of amino acids. Okay, they are the polymers of amino acids. And the polymers are held to each other by a specific bond called as peptide bonds. Okay. Now, from where does we get these proteins? We might have two kind of people who depends on the diet. Either it can be a vegetarian or it can be a non-vegetarian. So the basic the diet might contain the primary source, which is animal source, meat, milk, etc. Or if the person is vegetarian, then he might get from the cereals, pulses, nuts. Okay, so these all becomes the important sources of protein for the people who are vegetarian. And you might have seen people will be taking even supplements as well, the protein supplements or the amino acid supplements. So this becomes the source of the protein for any individual or the human. So this is a pictorial representation again. Coming to the per se, that is the digestion of the proteins. When we talk about the protein digestion, one important thing we have to always note, that is what is the role of cooking in the protein. Yes, we all know from our basics of the chemistry that cooking or the heat will cause denaturation. Okay, it will cause denaturation of the protein. So denaturation of the protein will increase the surface area. Okay, it increases the surface area so that the enzymes can act on it properly. So the digestion can occur very in simple way. Okay, the cooking will always help for the protein digestion. Now next comes is what is the role of the body? We have enzymes. In a broader aspects, the enzymes are proteolytic enzymes because we are digesting proteins, proteolytic enzymes. Most of these proteolytic enzymes are present as something called as inactive form. What form? We are present in inactive form, which are most commonly called as zymogens. Okay, if you go back to your very basics of enzymes chapter, there is a specific section called as zymogens. Zymogens are the enzymes which are inactive at the site of the production and they get activated only at the site of their function. What is the role of these zymogens? They are present because if they are activated within the cells, if they are activated within the organ or the tissue or the cell which is synthesized, it will lead to something called as autodigestion. Okay, the enzyme start digesting its own cell who is the synthesizer. Okay, so to prevent that, we are synthesizing the enzymes in inactive form, which we call as zymogens. Okay, most of the proteolytic enzymes are zymogens in nature. Okay, this much you should be very clear. Now, when it comes to proteolytic enzymes, there are two class of enzymes. One class, we call them as endopeptidases. Okay, what do you understand by endopeptidases? If you see a protein chain, okay, if you see a protein chain, amino terminal and the carboxy terminal, if the enzyme can act anywhere in between, okay, within the structure, the enzyme can act within the structure, anywhere it can go and cut the polypeptide chain. They are class of endopeptidases. Endo means within. Now, second class of enzymes includes exopeptidases. Okay, which class? Exopeptidases. What does they do? Same, if you take the polypeptide chain, amino terminal and carboxy terminal, they start digesting at particular end. Okay, either it will start at the amino end, we call them as aminopeptidases. Okay, if they start digesting from this end, carboxy end, then we call as carboxypeptidases. Okay, either they are starting from the amino end or they are starting from the carboxy end. If they are starting from the amino end, we call them as aminopeptidases. If it is starting from the carboxy end, we call them as the carboxypeptidases. These are the basic class of proteolytic enzymes which we come across in this section. Now, if we see specific places like digestion in mouth, does it happen in the mouth? No. Why it does not happen? Because there are no enzymes which are present in the mouth. Okay, there are no enzymes which can digest the proteins. So when you eat a food, when you are chewing, the bolus is formed in the mouth. There is no digestion of the protein. When it comes to carbohydrate, yes, it do. But here in proteins, there is no digestion in the mouth. Now the bolus has entered into the GI tract. Second place where it enters the stomach. In stomach, what happens? In stomach, we have specific enzymes which is secreted by in the gastric juice. Okay, in the gastric juice, we have specific enzymes which can digest the proteins. What are they? First, we have got pepsin, we have got renin, we have got gastricin, and also we have got the gelatinase. Okay, we have got the gelatinase enzyme. So these are the enzymes who will start digesting the protein. Okay, they will digest the protein in the stomach. Along with this, there is also role of HCl. Again, if you go back to the chemistry of amino acids and proteins are denatured by one, we have seen cooking. Second, by an acid. So in the stomach, again, an acid is there which will start denaturing the protein. It coagulates the protein. It brings the structure of the protein from quaternary, tertiary, secondary to the primary level. So that these enzymes, what enzymes? Either a carboxypeptidases, aminopeptidases, or the endopeptidases starts acting on anywhere of the structure. It will not start from any specific place, so that it takes a lot of time. It can start digesting anywhere. So it increases the surface area. So this is about the stomach. Now, once it is partly digested, now there is no complete digestion. It's a partly digested. Now, the partly digested food enters into duodenum. Okay. Who is there in the duodenum? Again, there are proteolytic enzymes in the duodenum secreted by or secreted along with the pancreatic juice. Okay, they are along with the pancreatic juice present in the stomach. Now it becomes alkaline. From acidic, the pancreatic juice converts it into alkaline structure. There are specific enzymes who will act, which includes tyrosine. Okay. Chymotrypsins, trypsin, carboxypeptidases, elastases, and collagenases. Okay, these are the enzymes who will start digesting the protein which has come in the duodenum. Okay, which has come in the duodenum. Now further it will go into the intestine, the fourth place, the small intestine. Now, till it reaches here, the protein has converted into smaller polypeptide chains. It can be even tripeptide or even dipeptide. Okay, the digestion is almost done now. There are enzymes in the small intestine also, like for example, aminopeptidases. Okay, we have enterokinase, prolidase, or tri and dipeptidases. So these starts digesting and completely makes into simple amino acids. Okay, simple amino acids, and we all know all the amino acids in the GI tract are alpha amino acids. Whatever we humans can digest and take into the body, or that is absorption, that is the alpha amino acids. So we have completely digested a protein into a amino acids. Now, these amino acids from the GI tract need to be absorbed. Okay, so that we'll see in the next slide. So now what we have done by digestion, we have converted the protein into simple amino acids, and these amino acids need to be absorbed. Okay, if we summarize the same thing, the dietary protein, when it reaches the stomach, okay, it reaches the stomach, it is denatured, partly hydrolyzed. In the intestine, they are converted into small peptides and amino acids. Now, these has to be absorbed. The absorption, is it active or inactive? It is active process. It's a active process. Means what? We are using the energy to absorb the amino acids. Okay, most commonly, either it is a carrier dependent or ATP sodium dependent pathway by which these are absorbed. Most commonly it is Symport mechanism. Okay, means two molecules are going together into the same direction. Okay, so we have completed up to this section. Now, is it important for us to understand the absorption? Yes. In the exam, we might expect a 5-mark question along with the dietary protein, that is the digestion. Okay, a digestion of a protein is a 5-mark question in the exam. So we might expect this as an exam question also. So when it comes to absorption, there are 5 different transporters. As I already told, it is dependent on transporters. So there are 5 different transporters who will be acting to absorb these amino acids which are digested from the proteins. Okay, what are they? First, transporter for basic amino acids. Okay, like for example, arginine, glycine. We have got a basic amino acid transporters. Next, for amino acid example, like for example, proline, we require specific transporter to transport this. Okay, proline. And also another amino acid, like for example, glycine. It helps absorption. Third, acidic amino acids. Yes, acidic amino acids. Acidic amino acids, like for example, glutamic acid, aspartic acid. They require specific transporters through which it can enter the cell. Fourth, like for example, if we have beta amino acids, all the amino acids are alpha. If we have beta, then there is a specific, like for example, beta alanine is absorbed only by beta transporter. Now, last one, what about neutral amino acids? Okay, neutral amino acids, like for example, alanine is there, leucine is there, isoleucine is there, valine is there, etc., etc. How they are absorbed? Neutral amino acids. The neutral amino acid absorption itself is very important exam question. Okay, we might expect a 3-mark question on this topic. That is the absorption of neutral amino acids. It is otherwise assisted. Okay, it is otherwise assisted by a tripeptide. You might guess that is the glutathione. The glutathione is a tripeptide. It's a tripeptide means it's again a protein which is containing 3 amino acids. What are the 3 amino acids? We have gamma, glutamyl, cysteine, glycine. Okay, it's made up of glutamic acid, cysteine, and glycine. So it is made up of 3 peptides, tripeptide, gamma, glutamyl, cysteine, glycine. So this molecule, in short form, we call it as GSH. Okay, this molecule has a role in absorption of neutral amino acids. So in the exam, they'll ask you for 3 marks. Write the role of glutathione in absorption of amino acids. Means what the examiner is trying to ask you or make you to represent on an answer sheet that how the glutathione helps in the absorption of neutral amino acids. Neutral amino acids. Now again, the glutathione is, as I told, it's a gamma glutamyl cystenyl glycine. This cycle is also called as Mr. cycle. Okay, in the exam they might ask you explain in detail about Mr. cycle. So what he is asking is to write about the neutral amino acid absorption, how it is absorbed by the intestine. Now, if we don't forget, where is the absorption? Absorption in the small intestine, because till it reaches the small intestine, the protein has become amino acid. In stomach, it is partially digested. In the duodenum, partially digested. Till it reaches the small intestine ileum, it becomes completely digested and becomes a single molecule, single unit of protein. What is it called as amino acids? These amino acids are absorbed by 5 different transporters: basic, amino, acidic, beta. Okay, basic, amino, acidic, beta amino acid, and last is the neutral amino acid. So we'll see how this neutral amino acid is absorbed. This is called as Mr. cycle. Okay, this Mr. cycle is basically with the help of one molecule which I mentioned, glutathione. The amino acid, which amino acid? The neutral amino acid which is present in the lumen need to be absorbed into the cell. In simple, we can see it can be absorbed like this, but it cannot. Okay, it cannot be absorbed that way. Okay, it cannot be absorbed that way. So to assist the same, the glutathione comes into picture. What does glutathione does? The glutathione, with the help of the enzyme transferase, gamma glutamyl transferase, takes the neutral amino acid and forms glutamyl amino acid by releasing cystenyl glycine. Okay, the gamma glutamyl cystenyl glycine has converted into glutamyl amino acid. So amino acid has bind with the glutamic acid. Cystenyl glycine are split into glycine and cysteine. Now, what is our main function? We want free amino acid into the cell so that it can be absorbed and transported in the blood. Now, glutamyl amino acid is split to form oxoproline and free amino acid. That is cyclotransferase. Now, we want glutamine back because we want the cycle to continue. We don't want it to stop somewhere. We want it to recycle. If we want to recycle, we have to resynthesize the molecule. How we are resynthesizing the molecule? What we have separated? 3 compounds: glutamic acid, cysteine, and glycine. Glutamine is there, cysteine is there, glycine is there. Again, rejoin them. Okay, you mix them, you join them. What you are doing? The glutamate is converted into glutamyl cysteine, means glutamic acid combining with the cysteine. When you join something, definitely we require energy. So what we burn? We burn ATP. Okay, which enzyme is here? Synthetase. When we are talking about the use of ATP, the synthase, we call them as synthetase. If the joining is happening without ATP, we call it as enzyme synthase. Here we are joining the compounds with the help of the energy ATP, so we call it as synthetase. So if you see in the picture also, glutamyl cysteine synthetase. Now, what is left behind is glycine. We want to add the glycine now. What we are doing? We are joining the glycine with the burning of energy. ATP is converted to ADP with the help of synthetase, that is glutathione synthetase, to synthesize the glutathione. So what is the final role? The amino acid, which was outside, is entering into the cell with the help of glutathione. The glutathione undergoes a cyclic manner where it is resynthesized. We are utilizing how many ATPs? If you see, 1 ATP, 2 ATP, and 3 ATP. Means what? For every one amino acid absorption, that is neutral amino acid absorption, we require 3 ATP plus 1 molecule of glutathione. Glutathione is recycled. Okay, it is recycled, so there's no problem with the glutathione. When it comes to ATP, yes, we are burning the energy. As I told, it's an active process. The absorption of amino acids is an active process. So we have completed the Mr. cycle where the amino acid is absorbed into the cell with utilization of the energy and a glutathione. Glutathione is a tripeptide, gamma glutamyl cystinyl glycine. Okay, please remember. And this is an important exam question which can be asked for 5 mark absorption, or only Mr. cycle can be a 3 mark question in the exam. So you should be able to write this detail in the exam with the structure. Okay, I hope till here we are very clear with the part of proteins. Now next we'll learn about what is the general metabolism of the amino acids. We can call it as protein in total because we are finally absorbing the amino acids, which is again converted back to the proteins. First, anabolic role. We all know proteins will build the body, and proteins are made up of amino acids. So, protein synthesis. Next, proteins will form a specialized compounds. Like for example, if you remember the enzymes are a majority are proteins. So they are synthesized from amino acids. So synthesis of amino acids, specialized compounds, which includes like, for example, heme part of the hemoglobin. Again, we require amino acids. The creatine phosphate synthesis, the creatine requires. The DNA and RNA, which is the purines and pyrimidines. Okay, purines and pyrimidines. They also require the amino acids. So in general, the amino acids or the proteins are required for these specialized compounds. If you catalyze a protein, when you break down a protein, what we get? We get the amino acids back. Okay, which can be again used for the protein synthesis. So it's a cyclical process we'll get. There are certain type of reactions which are helpful, where the amino acids are removed. If you go back to the chemistry of amino acids, we have something called as essential amino acids and non-essential amino acids. Essential are the one which need to be taken in the diet. Non-essential are the one which need not to be taken in the diet on regular basis. Why we are calling this? Because essential can be converted to non-essential, or essential amino acids will help in the synthesis of the non-essential amino acids. In them, one of the major reaction is transamination reaction. In further slides, we are going to see what is it when we are reading general reactions. But here, just to mention, this is one. Okay. Next, the carbon skeleton. If you see about the carbon skeleton, we have something like the structure of amino acids. If you remember, we have two functional groups, amino group and R group. When you remove this amino group, what is left behind is the carbon chain, which can be utilized for, like for example, you might have learned in gluconeogenesis, the proteins or amino acids will help in the synthesis of glucose. So this carbon skeleton can be helped in gluconeogenesis. Okay, they can be helpful in gluconeogenesis. Minor metabolisms like conjugating, okay, like conjugation process. If methyl group is there, methylation process, or sometimes you are adding amide group, amidation. Okay, so these are the things which can occur as a general. Okay, as a general metabolism, we have anabolic role, synthesis of specialized compounds, catabolic function. Okay, then transamination, which is special kind of reaction which we will be learning next. What happens to the carbon skeleton if we remove the ammonia? It can be utilized for gluconeogenesis. Other minor metabolisms include the conjugation, methylation, and amidation. So these are things which are general metabolism of a protein or amino acids. As I mentioned, there are specific reactions which are very much required for us to learn how the urea is synthesized. We are going to talk on urea specifically. One is called as transamination reaction. Second is deamination reaction. Third is trans-deamination reaction. Fourth, we are going to see about trans-methylation reaction. Fifth, we are going to see about decarboxylation reaction. So these are the general reactions which are required for us to understand how these are metabolized. Specifically for us for today, our main focus is these three reactions. What is required for today to understand how these things are working? One is transamination reaction, deamination reaction, trans-deamination reaction, trans-methylation, and decarboxylation reaction. Okay, we'll go to the first. First comes is the transamination reaction. As the name itself is telling, the transamination reaction is exchange of amino group, amino group of one amino acid. Okay, the amino group of one amino acid is donated to alpha keto acid to synthesize a new amino acid and a new keto acid. Okay, in other words, we are seeing the amino group of one amino acid is reacting with keto acid, alpha keto acid, to synthesize a new alpha keto acid and a new amino acid. Like for example, here in the example, if alanine reacts with pyruvate, we are going to get the glutamate. So one amino acid is converted into another amino acid. Specifically, if we remember, it should be always alpha amino group. Means the amino group which is attached to the alpha carbon. Alpha carbon is the first carbon to which both the functional groups are attached, carboxy group and amino group. So the transfer of amino group from one amino acid to the keto acid to synthesize a new keto acid and a new amino acid. So this kind of reactions are called as transamination reaction. For this, PLP, pyridoxal phosphate, is mandatory, and the class of enzymes are transaminases. Okay, like you might have heard about AST, ALT. So they all are transaminase enzymes who require the PLP. Okay, PLP as a coenzyme. Okay, partner to the enzyme. PLP. Okay, this is about the transamination reaction. Next, something called as deamination reaction. The name itself is telling, D means removal. The amino group is removed from the structure. Okay, the amino group is removed from the structure. What we are doing, like for example, NH3, COH, R group, H group. If you remove this, deaminate them. That is deamination reaction. There are two types. What are they? That is oxidative deamination and non-oxidative deamination. Okay, either it is oxidative or non-oxidative deamination reactions. We are not going to go in very detail for these kind of reactions. We'll stick to the oxidative because it is what is more important for us now. The oxidative, one of the example is removal of amino group from the glutamate. Okay, that is the oxidative pathway. Glutamate dehydrogenase. Okay, glutamate dehydrogenase will help in the reaction so that the glutamine will eliminate the ammonia. This reaction specifically occurs in two places, liver, and one more is the kidney. Liver, majorly it will be converted into urea. Kidney, the ammonia is released, which binds with acid to form ammonium, okay, which will neutralize the acid in the urine. So these are the two places where we find free ammonia. Otherwise, there is no free ammonia in the body. Okay, free ammonia is not seen anywhere in the body except at two places. One is the liver, other is the kidney. Liver, it will be utilized for the urea synthesis. In the kidney, it is utilized to neutralize the acid which is secreted in the urine. That is about the deamination reaction. Now, there is something called as third section. We term it as trans deamination reaction. As the name itself is telling, it is a combination of two. What are the two? One is transamination reaction. Second, deamination reaction. Both are happening. It's a combination of both. Why? Because in reality, whichever amino acid is undergoing transamination reaction finally end up having deamination reaction because we want to eliminate the ammonia. On daily basis, the ammonia is excreted in the body. It is removed. What is this reaction? It's a coupled reaction, but not at same place. Why? Because transamination occurs almost in all tissue. Deamination occurs specifically in the liver. Site specific. So by distance, by place, there are two different places, distinct from the place, but physiologically or chemically, the reactions are coupled or they are linked to each other. Like for example, here you can see the alpha amino acid is converted into glutamic acid. It can be anything. Okay, like for example, alanine it can be, or aspartic acid it can be. So any amino acid. Amino acid undergoing transamination reaction, forming glutamine or glutamic acid. This glutamic acid undergoes deamination reaction. Okay, deamination reaction with the help of glutamate dehydrogenase to form ammonia free plus alpha ketoglutarate. This alpha ketoglutarate, again, it will undergo same transamination reaction. So what is the end result? End result is free ammonia is released. So this kind of reaction is called as trans deamination reaction. It's a 5-mark question in the exam sometimes, or it can be a 3-mark question in the exam. So you have to explain both the transamination reaction and deamination reaction, and these both are distinct from physical nature. They don't occur at same place, but physiologically or by reaction, they are coupled. They are happening together. Okay, they are happening together in the reaction. That is about the trans deamination reaction. Okay. Next, we'll see something about the previous ones which we mentioned. That is transmethylation reaction. Transmethylation reactions are nothing but transfer of methyl group. Okay, there is transfer of methyl group from alpha amino acid. When it comes to decarboxylation reaction, we all know ammonia, carbon, carboxyl group, H and R. So this functional group, that is the carboxyl group, is removed as carbon dioxide. So decarboxylation reaction. So we can see these kind of reactions in general reactions of the amino acids. So here we are only focused on specifically transdeamination. To understand this, we should know what is transamination and what is deamination reactions. Now, these all things are happening. Okay. Now, what is this happening for? This is all happening to maintain something called as amino acid pool. Now, you can ask me, what is this amino acid pool? This is again a 3-mark question in the exam. Okay. What is this amino acid pool? The amino acid pool is nothing which has got any anatomical area, or it does not have any anatomical space which is occupied. But it is such a thing which is maintained in the body. Now, for example, we have influx who are contributing for the amino acid pool. We have outflux who is utilizing the amino acids from the body. Okay, these are two distinct things. So what are they? That is one, like for example, you have taken diet. Protein diet itself is the influx. It is digested to amino acid and it is contributing to the amino acid pool. Now, there is already a protein in the body. What we are doing? Protein breakdown. When you break the protein, what we are synthesizing? We are synthesizing again a amino acid. So it is again contributing to the amino acid pool. Is it only in? Obviously, we are using proteins or amino acids for some function. Like for example, we are using them for synthesis of plasma proteins. Very clear. Next, muscles. That is nothing but the body proteins. Obviously, some proteins are required for circulation. And last, disposal. Okay, disposal. There are two compounds which is disposed of. One is carbon skeleton, other is the ammonia. Ammonia is released as urea. Okay, so there is an influx, there is a diet and protein breakdown, which is contributing to the amino acid pool, which will increase the amino acid number, quantity. There is utilization, which will decrease the amino acid pool, that is plasma proteins, muscle synthesis, muscle protein synthesis, circulation, hormones, enzymes, okay, whatnot. Okay, then disposal. We are going to remove some of the amino acids from the body, final catabolism. In that process, carbon skeleton, we can use for glucose synthesis or any other molecules like pure intermediates. Ammonia, we have to remove it from the body because ammonia, one thing is typical. What is it? It is toxic. Ammonia is very toxic to the body. So that has to be neutralized. So how we are neutralizing? We are neutralizing by urea converting into non-toxic form. We are converting into a non-toxic form. Now, if you see here, this amino acid pool, is it static or dynamic? Definitely anyone would tell it's a dynamic because it's a continuous process. From the diet, we are taking protein, it's converted to amino acid pool. Breakdown of protein, it is contributing to amino acid pool. On other side, there is always continuous synthesis of plasma proteins, muscle building, circulatory hormones, proteins, enzymes, whatnot, and cell division, disposal of. That is the final degradation. So it's always a balance. There is something adding and there is always deletion. So the amino acid pool is always a dynamic state. It's a dynamic state of subject. Okay, so this is about the amino acid pool. Now next comes is final disposal of amino acid. We have seen amino acid when we dispose of, we get two compounds. One is leftover carbon skeleton. As I mentioned, this can be reused. But what about ammonia? Most of the time, ammonia cannot be reused. And second thing, it is very toxic to the body. Okay. Very little amount of this ammonia is also contributed from purine and pyrimidine bases. Okay. They both also will contribute to the ammonia. The major source of this ammonia is glutamic acid. Okay, the glutamic acid is also called as transporter form of amino acids for ammonia. Okay, glutamic acid is the major source. Then type of reactions we learned, deamination reactions. Okay, the deamination reactions. There are something called as modified carbohydrates, amino sugars. So when you break down the amino, we get ammonia. Okay, then there is something called as amine form of glutamic acid, glutamine. There is double amino groups, dibasic. So definitely if you remove one amino group, it will form the ammonia. Counterpart of glutamine is again asparagine. So these are the amino acids and the other compounds which are source of ammonia. Now, another 3-mark question. What is the fate? One major fate is, which we are going to learn, urea synthesis. But is it the only fate of ammonia? No. Like, same ammonia can be used for synthesis of glutamine from glutamic acid. Same ammonia can be used to synthesize asparagine from aspartic acid. Okay. Some free ammonia, as I mentioned, is excreted in urine. For what? As a buffer. As a buffer to neutralize the H plus ions. Acid. Neutralization of the acid in the urine. That is one of the mechanism of the kidney, how it will neutralize the urine acidity. That is by releasing the ammonia. Ammonia binds with H plus ion to form ammonium, giving ammonical nature for the urine. Okay. Now, if you see neatly, ammonia also maintains something called as pool, similar to amino acid pool. Okay. There are sources who will contribute to the ammonia. There is removal. That is the fate. So ammonia also is maintaining a normal level in the blood. Okay, it is not zero. Never. Ammonia is never a zero. Or ammonia is very high. It is maintained at a particular range. That is the pool, ammonia pool. There is sources and there is fate of the ammonia. Now, someone can ask, is ammonia toxic or non-toxic? Answer is always toxic. See, the nature is so beautiful. Anything which is toxic, the body has mechanism how it will neutralize it. Okay, it will neutralize it because the body don't want to die early. It want to live for eternity. It does not happen so, but still, because it has got its own programmed cell death, it knows it has to die, but still it has got the mechanism how it can have a healthy life. So the ammonia which is released from these amino acids is not released as ammonia. It is captured. It is made into non-toxic form. That is a urea. So before we go, the conversion of amino group or ammonia into urea, we'll see why ammonia is called, okay, why ammonia is toxic. What are the mechanisms? There are multiple mechanisms which are explained because there is no exact reason. Okay, there is no exact reason. So they have hypothesized some of the reasons, which includes first one, The ammonia, when the ammonia level starts increasing, what it does, it will utilize something called as alpha ketoglutarate. It will utilize alpha ketoglutarate. From where? It is from TCA cycle. Now, because of this, there is decrease in alpha ketoglutarate in the TCA cycle. TCA cycle goes slower rate, means less energy production. One, increase in ammonia will lead to decrease in energy synthesis in the brain tissue. Second, ammonia, ammonia will bind with glutamic acid to form glutamine in brain. What happens to glutamine? It goes out of the cell. It goes out. Now, what is reducing? Reduced glutamic acid. Now, one of the important function of glutamic acid in the brain is formation of a neurotransmitter GABA. Now, GABA synthesis is also reduced. There is a decrease in GABA production. Okay. Now, third, the same thing, glutamine is osmotically active. Again, it will damage the brain tissue. Okay. Fourth, the increase in this glutamine. Okay, increase in outflow of glutamine for exchange of tryptophan. Okay, for the exchange of tryptophan. Now, what does the tryptophan will do? It will synthesize its neurotransmitter, serotonin. There is increase in serotonin, decrease in GABA, increase in osmolality, and decrease in energy production. So what happens? Overall, low energy, increase osmotic pressure, and derangement in neurotransmitters. So these all will contribute because of what? Because of high ammonia, which is toxic to brain. Okay, this is toxic to brain. So now you tell me, do you want the ammonia to be increased? No, we don't want it to increase. So what is the best method by which it is neutralized? Urea production. So this is the reason why we have to learn something on urea cycle. Okay, the basis for urea cycle. What is the role of urea cycle in this mechanism? How it is capturing the ammonia, neutralizing it, and removing it from the body. Okay, so now we'll talk on urea cycle. If you see about the urea cycle, very beautiful cycle. Every exam you can expect long essay. And now we have something on case-based questions. Okay, case-based pattern also you might expect one question on urea cycle. Or sometimes only some part they might ask for 5 marks. Or sometimes they might ask you some enzyme disorders or something like that, energetics, which is for 3 marks. So you can imagine how important is the urea synthesis. Otherwise, this is the mechanism by which ammonia is disposed of. How ammonia is disposed of is by urea cycle. Now we'll see some features of this urea cycle. If, like, imagine this is a 10-mark question in the exam and you have to explain the urea cycle. First, always write what are the other names. Urea cycle is simple one name, that's good. But when it was identified, It was identified by Krebs and
Hensley.
So it is called as Krebs-Hensley cycle. One more thing in this is an important compound called as ornithine. So it's also called as ornithine cycle. And one more thing, it has role along with the TCA cycle. So it's called as urea bicycle because it is related to even TCA cycle. So it's linked with the TCA cycle. It is having connection with the new TCA. So these are the other names. What is the purpose? We are very clear on this purpose. We want to neutralize this. Okay, neutralize this ammonia. Now, what is the site? I already mentioned liver. In the liver, two places these reactions occurs. One is mitochondria, other is cytoplasm. Okay, because some enzymes of the urea cycle are present in the mitochondria and some are present in the cytoplasm. So it occurs at two sites. The total reaction occurs at two sites. Now, urea contains two nitrogen, of which one is the free ammonia and other is from aspartate or aspartic acid. One is derived from ammonia, which is glutamic acid. Second one is derived from the aspartic acid, which is in the reaction. Okay. What are the substrates? Obviously, one is ammonia by itself. Okay. And second is carbon dioxide. Okay, these are the two important compounds which are required for urea synthesis. Substrates. And what is our end product? Not to tell again. End product is urea. So we have the substrate, we have the end product. Now, what is missing in between this is the steps. Steps. The most simplest cycle in whole biochemistry is the urea cycle. Okay, I can bet on it. Why? Because it's very simple. 5 steps you can count on your fingers. What are these 5 steps? We are going to see in specific pathway itself. Okay, now if I tell you the 5 steps also, it's very difficult to remember. So there are only 5 steps. Now we'll see one by one how it is synthesized. Okay, coming to here, we have, I told you what is the substrate required. The substrate required is ammonia plus carbon dioxide. Ammonia plus carbon dioxide, and also we might require some amount of water. When it is condensed, the ammonia is condensed with carbon dioxide, we get something called as carbamoyl phosphate. From where is this phosphate coming? Definitely, phosphate is donated by one of our important compound, ATP. Okay, the ATP will donate the phosphate group. How many ATPs are utilized? 2. So we get 2 ADP plus PI. Now the carbamoyl phosphate is synthesized. Now what is the enzyme? That is synthetase. Why we are calling synthetase? Because we are utilizing ATP. What is the full name? That is the product name. Okay. Carbamoyl phosphate synthetase 1. Okay, there is one more counterpart for the same, which is carbamoyl phosphate synthetase 2, which is present in cytoplasm. Now you remember, this reaction is happening in mitochondria. Okay, the reaction is happening in the mitochondria. We have synthesized carbamoyl phosphate. Now we want this reaction to further continue. Now who is coming in is ornithine. Okay, the ornithine is present in cytoplasm. Okay, the above section, M stands for mitochondria and C stands for the cytoplasmic reaction, because enzymes are present in cytoplasm, some enzymes are present in mitochondria. Ornithine is present in cytoplasm. It is transported into the mitochondria, where the ornithine combines with carbamyl phosphate to synthesize something called as citrulline. Okay, carbamyl phosphate binding with ornithine to synthesize citrulline. Citrulline is synthesized in the mitochondria, then again it will be transported into the cytoplasm. Okay, ornithine is present in cytoplasm, transported to mitochondria, combines with carbamyl phosphate, and the compound citrulline enters into the cytoplasm for further reactions. Who is the enzyme? That is a ornithine transfer, that is trans. What we are transferring? Carbamylase. So trans carbamylase is the enzyme. In the citrulline, in the cytoplasm, is converted into argininosuccinate. Now you can imagine how it is happening. Citrulline without binding with someone, it is happening? No. Who is coming into the picture? As I told, aspartate or aspartic acid. Okay, aspartate will bind with citrulline to synthesize argininosuccinate. Argininosuccinate. Again, we are utilizing ATP. Okay, ATP is converted to AMP plus PPI, which is equivalent to utilization of 2 ATPs. Okay, which is equivalent to utilization of 2 ATPs. Again, enzyme is synthetase, argininosuccinate synthetase. Okay, now this is converted into or split into arginine and fumarate or fumaric acid. Now further, arginine is converted into ornithine back with release of urea. So we have synthesized our urea, we are lysing it, so it is arginase with the utilization of water molecule. A water molecule is utilized to split urea and we get ornithine back. So if we see the reaction, the ornithine acts like a catalyst, enters the reaction, takes part in the reaction, and it comes out without undergoing any change in its structure, not even single carbon. Okay, a single carbon is also not changed in the ornithine. That's why the urea cycle I told you, it's also called as ornithine cycle. Okay, it's also called as ornithine cycle because ornithine plays as an important compound under which this whole urea cycle is dependent, and ornithine acts like a catalyst. Without undergoing any change in itself, it completes the reaction. Now here if you see the fumarate, it can enter the TCA cycle and gives back the aspartate. So I told you one more thing, it's called as urea TCA cycle or urea bicycle because it is linked with one more cycle which is happening in the mitochondria. Okay, this is also happening in the mitochondria. So it is connected to the TCA cycle. The urea cycle is connected to the TCA cycle. That's why it's called as bicycle. So ornithine cycle or urea bicycle, or it is also called as Krebs-Henselt cycle because they were the founder who have identified the urea cycle. Okay, so this is about the urea cycle. Again, to summarize the same, we have ammonia binding with the carbon dioxide and water to synthesize carbamyl phosphate in the enzyme synthetase with the utilization of 2 ATPs. And then ornithine enters the mitochondria, okay, through the transporter. It binds with carbamyl phosphate to synthesize citrulline, which comes into the cytoplasm. Citrulline further condenses with aspartic acid or aspartate to synthesize argininosuccinate, utilization of the energy, which is equivalent to 2 ATPs. With the enzyme synthetase, which synthetase? Argininosuccinate synthetase. Further, argininosuccinate is split into 2 compounds. One compound is nothing but the arginine, the half section. Arginine further undergoes reaction with the help of water, hydrolysis. We are releasing the urea molecule, which is water soluble, and we are resynthesizing or synthesizing back our ornithine. On other side, the fumarate, which is released, it can enter the TCA cycle, converted into aspartic acid, and it can again contribute to the urea cycle. So two compounds are contributing to the urea cycle. One is aspartic acid, which is from the TCA, and the ornithine, which is resynthesized back. So this is about the urea cycle. Okay. Now, if you write the urea cycle, is it fine? No. You have to talk about the energetics. If you see overall reaction, okay, another 15 minutes will complete the section. So if we see the overall reaction, ammonia binding with carbon dioxide plus aspartic acid will synthesize urea plus fumarate. We have utilized 4 high energy compounds. The fumarate, okay, the fumarate to aspartate synthesizes 1 NADH. So 1 NADH can give 2.5 ATP. So what is the net? 4 are utilized, 2.5 are added. Net is minus 1.5 ATP. So urea cycle is a catabolic reaction where we are expendituring the amount of energy. How much energy is expenditured per urea cycle? Per 1 turn. Okay, per turn of urea cycle, we are expendituring 1.5 ATPs in the body. Okay, per cycle, per ammonia removal, per urea cycle, we are utilizing around 1.5 ATPs. Okay, this is about the energetics. Now, what about regulation? There are 3 sets of regulation. One is called as coarse regulation, other is fine regulation. Okay, other is called as compartmentalization. Okay, coarse regulation, like for example, if the person takes diet, protein rich diet, ammonia synthesis increases, urea also increases. If person is starving, because in starving what happens? The catabolism of protein occurs. So catabolism of protein will lead to increase ammonia, again increase in urea levels. So that is coarse regulation. Fine regulation is by modulating something called as carbamoyl phosphate synthase 1 or synthetase 1 enzyme by a molecule called as NAG. Okay, there is a N-acetyl glutamine. Okay, N-acetyl glutamine. So N-acetyl glutamine, if there is high in NAG, increase in carbamoyl phosphate synthetase activity, increase in urea. If NAG is low, urea is also low, synthesis is low. That is a fine regulation, very fine. Other is compartmentalization. Why we are telling compartmentalization? Because some enzymes are present in mitochondria, some enzymes are present in cytoplasm. If you remember the regulation of enzymes in the enzymes chapter, compartmentalization is also a type of regulation. Okay, it's a type of regulation. So this is again a 3-mark question in the exam. Total urea cycle plus energetics plus regulation can be a 10-mark question. Okay, be very clear on this. Now, last section comes is the what are the errors of metabolism. Now, we all know when A is converted to B, we require enzyme 1. When B is converted to C, we require enzyme 2. When C is converted to D, we require enzyme 3. Imagine enzyme 1 is absent. Now, do you think A can be converted to B? No. What happens? A starts increasing. Now, imagine enzyme 3 is absent. What is happening? C is increasing, Same holds good with the B and C, feedback inhibition, kind of feedback inhibition, where there is accumulation of the substrates which are prior to the reaction. The enzyme deficiency is inherited most commonly. Okay, that's why we call it as inborn errors associated with the urea cycle. There are few. Okay, there are few inborn errors which are associated with the urea cycle. What are they? If you see in general pathway, okay, the ammonia binding with carbon dioxide with water by enzyme carbonyl phosphate synthetase 1. If this is absent, what is increasing? Ammonia. What is the disorder? Hyper ammoniemia type 1. Clear? Now, ornithine binding with carbonyl phosphate by trans carbamylase. Okay, by trans carbamylase. If this is absent, what is accumulated? Again, ammonia, carbonyl phosphate. That is hyper ammoniemia type 2. Now, up to citrulline is fine. Now, argininosuccinate synthetase. This is absent. What happens? Citrulline increases. What is the disorder? Citrullinemia. Up to argininosuccinate is fine. Now, enzyme lyase is absent. Who is increasing? Arginosuccinate, which is nothing but arginosuccinic acid. Okay, argininosuccinic acid, because it is water soluble and excreted in urine, it is also called as acid urea. Now, last step, arginase is absent. What happens? Arginine starts increasing. So we call it as hyperarginemia. Okay, so this is the overall structure of the pathway. Okay, this is the overall structure of the pathway. Now we'll see the last section individually. We have first disorder, hyperammonemia type 1, type 2, citrullinemia, arginosuccinic aciduria, and last is hyperammonemia, argininemia. Please remember, this is a 5-mark question in the exam. Sometimes they'll ask you to write the enzyme absent in the each thing. Okay, please, I have prepared this specifically for this session into a single format, how it can be easily understood. First, hyperarginemia type 1, carbonyl phosphate synthetase 1 is absent. Hyperammonemia type 2, it is ornithine transcarbamylase enzyme absent. Third, citrullinemia, again argininosuccinate synthetase absent. Here lyase is absent. Here arginase is absent. Okay. Now inheritance. I told you inborn errors are all inherited disorders. Out of all these, type 2 is X-linked. All other are recessive, autosomal recessive. Okay, AR, autosomal recessive, autosomal recessive. All are autosomal recessive except type 2 hyperammonemia, which is X-linked, means even mother will have the features. Okay, if the mother is fed with more amount of protein, what she'll end up is, she'll also end up having the hyperammonemia, increase in ammonia levels. What are the key clinical features? Definitely, there is increase in ammonia and mental retardation. That is a key feature of this. And also, because ammonia is increased, toxicity of the ammonia features. Okay, ammonia toxicity happens. Very severe. In second, ammonia is increased, not so much like type 1, and little bit of toxicity, no mental retardation. Citrullinemia, again here, very high ammonia with toxicity, okay, with toxic features, toxic symptoms, and also mental retardation. Arginosuccinic acid urea, it happens usually before 2 years and it is very fatal. The child might not survive. Last one, hyperargininemia, simple increase in the ammonia levels. If you do blood investigation, urine investigation, and CSF, do you see these things? Yes. Increase in ammonia levels, increase in ammonia levels, and also ornithine also, because ornithine is not utilized by the reaction. When it comes to citrulline, again increase in ammonia, increase in citrulline because citrulline is getting accumulated, and the same citrulline is excreted in the urine. Large quantity is excreted in the urine. Next, same thing, argininosuccinic acid urea. Again, increase in blood, increase in urine excretion, the acids. Last one, that is increase in blood CSF arginine. If you stop feeding the arginine, definitely the child or the person will have the less symptoms for the same. How about incidence? This is 1 in 1 lakh, 1 in 2 lakh. Same holds good for the citrullinemia, 1 in 2 lakh. Arginic acid urea, 3 in 2 lakhs. And last one, hyperargininemia, 1 in 1 lakh. So these are the features which is most commonly asked for 5 marks. Enzyme defect, what is the inheritance? All these, type 2 is only X-linked, all are autosomal recessive. Clinical feature, mental retardation is very common in hyperammonemia type 1 and citrullinemia. Very fatal, type 1 and arginosuccinic acid urea. Okay, incidence, as I mentioned. Okay, so these are the things which are related to the disorders of the urea cycle or urea cycle inborn errors of disorders. So last section is some clinical feature or normal values. Now I told you ammonia is maintained in a pool. The normal value is less than 50 micrograms per DL. Okay, if it goes more than that, hyperammonemia. I told you ammonia is toxic to brain or brain cells. So that's why we have to maintain it in normal. How it is maintained? It is synthesizing urea. Now urea is present at 2 places, blood and urine. Obviously, it's a water soluble. Excretory route is majorly urine. In blood, what is the normal level? 20 to 40 milligram per DL or deciliter. Same thing is excreted in the urine per day, which is 15 to 30 grams per day urine output. Okay, per day urine output. So this is the features. Now one more feature of urea is it's an important indicator of renal function. Okay, we can use the urea as important indicator for the renal function in concordance or in combination with creatinine. Okay, you might have known what is creatinine. So in combination with the creatinine, the urea is used for detecting or identifying the renal function. Okay, so with this, we will be ending with this section. To summarize, we have seen first general features of proteins. Second, we have seen about general reactions of protein. Third, we have understood how proteins is converted into amino acids and how amino acids are absorbed. Okay, absorption of amino acid, specifically Meester cycle. Fourth, What is ammonia? How it is toxic? How it is neutralized? Yes, we have learned. Specifically in the neutralization, we have learned about urea cycle per se. Okay, steps, energetics, regulation, and we have learned about the IEM associated. That is the inborn errors of metabolism associated with the urea cycle. Okay, and last we have learned something about the what are the key features or the clinical features, or what is the clinical relevance, significance. Okay, what is the clinical significance of learning the normal values of the ammonia and normal value of the urea. Okay, so at the last section, I would like to just have a note. After learning all this, what you can expect as a question. Okay, that is the most commonly asked university question. First, like for example, you can see here, write in detail how ammonia is detoxified. In simple, he is asking you, write about the urea cycle with its energetics, how much energy is utilized, how it is regulated, as we learned, fine regulation, coarse regulation, and the compartmentalization. Next, what are the errors of associated? You can expect this kind of question for 15 mark or 10 mark. You can see how it is split. Okay, this is first. Second, you might expect how the proteins are digested and absorbed. Again, you have to write neatly what are the enzymes, stomach, duodenum, intestine, absorption 5 transporters, special note on glutathione or the Mr. cycle. Third, trans-deamination reaction. This can be 5 mark or 3 mark. You have to explain how transamination occurs, deamination occurs, and what is trans-deamination reaction. Fate of ammonia is for 3 mark. You can write what are the different fates of ammonia. Even sometimes they'll ask you nowadays, what are the normal levels of urea in blood, how much it is excreted in urine, and they might ask you about ammonia also. Okay. Now, last one, they might ask how the neutral amino acids are absorbed. That is, Mr. cycle or role of glutathione in absorption of neutral amino acids. Okay, this can be again a 5 mark question or 3 mark question you can expect in the university exams. So in this section, we have learned all these things. Okay, so when we go to the exam, we always make sure that we know what is the questions, because unless you don't know the question, you don't know what can be the answer for it. When you are reading the book, you should always make sure you know question. Once you know the question, you know what should be the answer. And when you know the answer, then you will be very confident to attempt in the exam and you will not be fumbled around. Okay. So with this, I will conclude for today, which we have seen the